Good afternoon, all, and good morning to our international speakers in Canada. It's a great pleasure to be back here in the second part of our first event about biological aspects of early life adversities. Claudia, Deborah, and I will introduce our speakers who will give a 20-minute talk each, and then we will have a moment for questions and discussions. So if you have any questions, please leave them in the chat, and then we'll pass them on to our speakers. To start this session, Claudia will introduce our first speaker, Professor Andrea. Claudia, please.
Good afternoon, everyone. It's a great pleasure to me to introduce to you Dr. Andrea Jakowski. She's Associate Professor at the Department of Psychiatry in our university in UFESP and the coordinator of the graduate program in Psychiatry and Medical Psychology. She has been studying for a long time the effects of early life stress exposure on brain development and psychopathology. Please, Dr. Jakowski.
Much, Professor Claudia Berlin. I would like to thank you everybody for the invitation for this meeting. And I would start my presentation. Can you all see my presentation? So I'll be talking this afternoon with you about a project that we've been working on called Maternal Adversity, Inflammation, and Neurodevelopment, how intergenerational processes perpetuate disadvantage in a low resource setting. So first, I would like to acknowledge FAPESP and NIH for a joint initiative of funding this project and CNPq. Well, intergenerational trauma is the transmission of an environmental adversity effect from one generation to the other. And this concept of intergenerational trauma was first introduced in the psychiatric literature through the descriptions of behavior and clinical problems in offsprings of Holocaust survivors. So the offsprings, they were not exposed to the adverse condition, but for some reason, they had more psychiatric problems than the average population. So that's how initially the psychiatrist starts studying the transmission of the trauma from one generation to another. And in the initial focus, the initial biological focus was on the HPA axis. The preliminary or initial hypothesis regarding the Holocaust offspring was that the parental experience may alter the regulation of the stress-related pathways early in the development. Well, we know that early in the development, the HPA axis is very susceptible and it is vulnerable to environmental perturbations. What else did we learn from that? Well, the parental experience, they might alter the regulation of the stress-related pathways early in the development, and we know for sure from the literature that this regulation of stress neurocircuitry, it's a fundamental feature of mood and anxiety disorders, also including PTSD, and they were found to be prevalent in the offsprings of the Holocaust survivors. And moreover, we know that low cortisol and increased glucocorticoid receptor sensitivity in Holocaust survivors were also observed and in other trauma-exposed individuals with PTSD. So that's how the initial biological studies were conducted, trying to explain how the trauma is transmitted from one generation to the other. And there has been many proposed mechanisms trying to explain how the adverse childhood experiences, or ACEs, they are transmitted from one generation to the other. One proposed mechanisms from that is that the ACEs influence the parenting behavior, affected the parent-child interactions, and also increasing the risk of a child to encounter additional adversities. But we cannot forget there are also genetics mechanisms behind that predispose towards the individual towards an experience in ACEs, or even that these genetics predispositions is shared between parent and child. Epigenetics also play an important role in this transmission because adversities, they lead to epigenetics modification, and these modifications, they are passed from parent to child. And more recently, the literature has been focusing on fetal programming. What is the fetal programming? Well, we know that the maternal adversity somehow is communicated to the offspring via the maternal-fetal transmission. And the communication between the mother and the fetal is through the placenta. We know that the biochemical changes that occur in the mother arising from adversity, they will influence the fetal neurodevelopment. And these transmissions that occurs during pregnancy. So I'm going to show you some of the proposed pathways, and then we're going to jump to the study that I would like to present to you this afternoon. Well, we know we are talking about adverse childhood experience that happened with this pregnant woman that would alter, the hypothesis is that will alter the HPA axis dysfunction that will be transmitted to the fetus and will cause an alteration in the cortical limbic circuits that later on will lead to an emotional dysregulation that can be measured at 12 and 24 months. But it's not only that. We know, and there has been studies showing that these ACEs experience, they would increase maternal inflammation that will also be transmitted to the fetus by crossing the placenta, and will alter the dopaminergic neurons, leading to impulsivity later on in life. So we are talking about events that happen early in maternal life that will affect the brain development of the offspring. But it's hard to isolate only these effects of the adverse experience that happen during childhood, because we do have additional prenatal influences. We do have genetic influences. We do have recent stress or adversity that this mother may be suffering during pregnancy or at her life. And we do have the maternal health also, that we cannot exclude these effects. And we cannot exclude also the postnatal influences that might change the baby's brain circuitry and lead to emotional dysregulation at 24 months, or increasing impulsivity that might lead to externalizing behavior later on in life. We need to be evaluating also parental and child interaction, the child's health, and the child's own adverse experiences. Well, there has been a couple of studies that had studied the effect of ACEs, the increase in inflammation, and the impact of ACEs and inflammation in brain development. In 2016, there had been a huge meta-analysis that had associated ACEs with elevated levels of C-reactive protein, a study that has been conducted in more than 16,000 adults. Alterations in inflammatory markers like interleukin-6 and TNF-alpha also, and some imaging study that has shown that increased maternal inflammation is associated with altered connectivity between cognitive control brain regions and their offspring. So there is evidence in the literature. And moreover, there has been preclinical studies that associated stress, inflammation, and placental gene regulation. So there has been a couple of studies in the literature, but there hasn't been a study in Brazil. And why conduct a study and a research in Brazil? Well, Brazil, it's a very large country. We have more than 209 million inhabitants, and the country is marked by diversity in regional, ethnic, cultural, and socioeconomic disparities. When we talk about disparities, the inequalities rates in Brazil are very high. Before the pandemics, more than 23 million people live in Brazil below the poverty line. That means a monthly income less than 232 reais a month. And we know that it rose a lot in the last 4 years, and mainly now during the pandemics, increased a lot, the disparity in Brazil. According to the World Health Organization, Brazil has one of the highest rates of homicide in the world, and it changes throughout the Brazil. When we look at the map, we can see that the rates also of homicide, they change from one region to the other. And why did you select to conduct this study in São Paulo? Well, São Paulo is the largest city in Brazil and the largest city in the Americas, also in the southern hemisphere. The number of inhabitants of São Paulo is more than 12 million inhabitants, over 44 million in the state. And we are narrowing down. Why in São Paulo and now why in Guarulhos? Guarulhos is the second largest city in São Paulo, is where our international airport is, and the inhabitants is over 1 million inhabitants estimated more than in 2019. And also the rates of violence are extremely high. And there's more reasons why we chose to conduct this study in Brazil. And one of the main reasons despite the inequalities is because of our universal healthcare system called the SUS. Brazil has the largest universal healthcare system. SUS provides healthcare to the population, is highly utilized, and is often the only source of healthcare for those living in very, very low-income areas or regions with high vulnerability. In 2006, SUS was responsible for about 98.5% of all pregnant women that were receiving prenatal care, free of any charge. And these primary care clinics that are called UBS, basic health units, they are very close to their homes and linked to specific hospitals from the public system. So in our study, we have three main aims. First is to identify the infants, identify and influence the influence of maternal ACEs on the child's emerging cognitive control. What is the hypothesis behind that, and how are we going to do that? Well, our hypothesis is that the maternal ACEs will be associated with the brain. The offspring will have reduced volume and functional connectivity in the cognitive control brain regions. How are we going to do that? Performing a brain MRI when the newborn is between 2 and 4 weeks old. We also hypothesize the maternal ACEs will be associated with decrements in emerging cognitive control behavior at 12 and 24 months. And how are we going to measure that? We're going to be applying the lab temperament assessor and battery when the infant is 12 and 24 months. Our second aim is to detect the influences of maternal ACEs on prenatal inflammation and on placental DNA methylation and gene expression. The hypothesis behind that is that the maternal ACEs will be associated with elevated maternal inflammatory markers, for example, IL-6 and CRP, IL-6 beyond other interleukins that will be evaluated. Moreover, prenatal inflammatory markers will be associated with reduced methylation of placental immune activated genes with a corresponding increase in transcription of these genes. And our third aim is an aim of establishing pathways, trying to study mechanisms of transmission of the trauma from the mother to the offspring, and how we'll be studying them, so trying to establish pathways by which maternal ACEs will influence the infant emerging cognitive control behaviors. And the hypothesis is that the association between the maternal ACEs and the offspring outcomes, being brain circuitry or brain volume and behavior measured by lab tab, will be mediated by the reduced methylation and the increased expression of immune genes, activating genes on the placenta. Of course, as I mentioned to you earlier, we cannot exclude other influences when we have both prenatal and postnatal influence. So prenatal influence, we're going to be genotyping these mothers and these children and evaluating polygenic risk scores. We'll be evaluating as well recent stress and adversity, as well as maternal mental health by clinical assessment. As for postnatal influence, we'll evaluate parent and child interaction by measuring it during the MRI visit and a home visit, the child health based on pediatric records, and the child own adverse experiences using clinical assessment. So how this study will be conducted, actually is being conducted. So our approach initially focused on women and their offspring that received prenatal care in these prenatal care units, that clinics that call UBS in Guarulhos. But because of the pandemics, we have to include other places in São Paulo with very similar profile of the population. So in Guarulhos, we have a network of 69 clinics that follow about 70 pregnant women monthly. And the greatest number of births, about 35% of the total number of births from Guarulhos occurs in this maternity. That is the maternity that we work with. So for this study, we enroll pregnant women at the third trimester, aged from 18 to 38, with single naturally conceived pregnancies, a total of 580 women, 290 that had no or low ACEs, and 209 that were exposed to adverse childhood experience during pregnancy. So we assessed all the ACEs, and we collected a blood sample and a hair sample for inflammatory markers and for cortisol analysis. And at delivery, we collect biospecimens like as placenta, maternal peripheral blood, and cord blood of a total number of 400. So that's just an example of how the prenatal card is in Brazil. So each woman have a prenatal card and has at least 7 prenatal appointments. So all the information regarding number of gestations and all socioeconomics and antecedents we collect from this card. Postnatally, when the baby is from 2 to 4 weeks old, we perform a brain MRI after a pediatric evaluation. So we scan babies sleeping, non-sedating after they are fed. We also collected origin from these babies and from this mother, saliva from mother and baby for cortisol and oxytocin analysis. We do the recording of the mother and baby interaction using the CIB, the coding interacting behavior, and also we apply some maternal clinical scales. This slide was supposed to be before, just to show you that during delivery, we collect maternal blood, we have cord blood collection, and we have placenta samples collection. So 2 samples of placenta from the fetal side from each of these parts, a total of 8 fragments of placenta. So also postnatally, after the MRI, at 6 and at 8 months, we do a follow-up. We collect saliva from the diets from mother and baby for cortisol and oxytocin analysis. We record the mother and baby interaction using the CIB, and we continue performing maternal and paternal clinical evaluation. At 12 and 24 months follow-up, we collect again the saliva from the diets, repeat the CIB. We do the lab tab, so the laboratory temperament assessment battery, to evaluate stress situations and other everyday situations that simulate real daily situations, and we can reliably observe individual difference in the expression of emotion. We'll do the Bailey to evaluate the global development and continue the maternal and clinical evaluation. So that's This table just illustrates our experience. So doing the ACEs evaluation at 3rd trimester and blood sample. At birth, we collect the placenta. We also, at 2 weeks old, we collect the MRI scan and clinical scales, and then we continue doing that until following up the baby until the baby is 24 months. So before we got funded, we did a pilot study with 62 pregnant women starting following them at 3rd trimester, using the child trauma questionnaire to evaluate childhood trauma and mini to evaluate psychiatric conditions. And we're able to collect maternal head for 58 mothers. We collect biospecimens from delivery for more than 40 mothers. And we perform MRI in over 40 babies during natural sleep and collected hair sample. Well, preliminary results from the pilot sample show us that there was a significant relationship between maternal childhood trauma and reduced connectivity, functional connectivity in the offspring, in prefrontal cortex and other brain areas involved with cognitive controls, which corroborates our preliminary hypothesis. Moreover, the mothers which had trauma, we had ACEs, they do demonstrate an increased levels of hair cortisol compared to the ones that were not exposed to ACEs during childhood. So now we started our project. So our project was funded in 2020, and unfortunately, because of the pandemics, we had to delay a little bit the data collection, but we started last December, and so far we were able to recruit 106 women in the study, include actually 106 women in this study. We were able to collect hair samples from all of them, and maternal and blood from 97. We had 80 babies that were born so far. In terms of biological samples from delivery, we have more than 50 placentas, maternal blood, and almost 50 cord bloods. So during MRI, we were able to perform successfully so far 42 MRIs. We have origin collected from 43 dyads, 43 baby hair samples, 34 saliva from babies and 16 from mothers for oxytocin and cortisol. And here is just an example on how we do the recording of mother and baby interaction using the CIB. We did that already for 43 subjects. So next step, we will continue the recruitment, and we are going to start conducting the 6 and the 12 months follow-up. So the team is huge. In order to make this project happen here in Brazil, I would like to thank you, all the PIs and all the research associates, the collaborators, Dr. Claudia Berlin, all the PhD and the postdocs, masters, graduate students, pediatricians, and mainly our study coordinator, our field team that are in charge of doing all the recruitment at the UBS level. I'd like to thank everybody, the technicians, and also like to thank the US team, Jonathan Pausner and Cristiane Duarte, and mainly they are the PIs on the R01 grant that we were awarded together. Acknowledge again FAPESP, CNPq, Fulbright, CAPES, and FIDI for supporting our study, and thank you all for your attention.
Oh, this is really interesting. But let's continue, and we'll have a discussion at the end. So it is my great pleasure to introduce you Professor Dominique Walker. She is a professor at the Department of Psychiatry at McGill University in Montreal, Canada. And I had the pleasure to first meet Professor Walker when we were both postdoc students in California. I was working with Seymour Levine in Stanford University, and Professor Walker was working with Mary Dahlman in the University of San Francisco. So we had some meetings, dinner lab meetings, which were really fun at the time. So Professor Walker has contributed to the field of early life stress and has published more than 100 papers in this field. Her most recent research grants involve the study of early life adversity on circadian rhythms, metabolism, and the development of the amygdala prefrontal cortex circuit, which is the topic of her lecture today. She is a member of several important scientific societies and was recently elected Secretary of the Pan American Neuroendocrine Society. Without further ado, I invite Professor Walker to give her talk, Early Life Stress and Development of the Corticolimbic Circuit in the Rat.
Thank you, Deborah. Thank you very much for this very nice and kind introduction. So I'm going to try to share my screen now. Let's hope it works. Okay. It's not working, right?
Working.
Okay, let's try again. No. Okay. So I hope the screen sharing works. So thank you very much for your very kind invitation. It's a real pleasure to be part of this mini symposium. And I will try to share some of the recent work we've been doing to look at the effect of early life stress on development of the corticolimbic circuits. And those are preclinical studies that we've performed in the rats. As you know, and as a previous speaker already presented, early adversity has long-term consequences and even transgenerational consequences on brain development, on many responses to stress and different behaviors that are derived from it. And those consequences arise from a stressful life environment, whether prenatally or postnatally. This leads to a number of DNA changes that Andrea has been talking about, and the next speaker will be talking about even more about the epigenetic changes. Neural circuit changes impair brain development and, in fact, effect on stress responses, in particular exaggerated stress responses. This leads to an increased risk of cognitive deficits, socio-emotional deficits, and anxiety behavior. Why we're looking at very early development is that there are very different critical windows of sensitivity that occur during development, and in particular with respect to the development of specific systems. The earlier vulnerability window is probably for all the somatosensory senses, vision, hearing, and touch, and plasticity in the amygdala-dependent learning, as you see here on the red curve, follows very closely, and this happening in very early period of life. Followed up after that for everything that has to do with hippocampus-dependent learning, with different waves of plasticity as a function of the modality. And then higher cognitive functions, as we know that in humans, those develop later. And in fact, in rodents, there's also the development of prefrontal function is...
Excuse me, Dominique, can I interrupt you just a moment? Can
Yes.
you put your presentation in presentation mode? Because we are looking at all the slides.
Oh, it is in presentation mode in my, on my... Okay, so I'm going to stop sharing and then try it again.
I'm sorry.
No, that's okay. That's okay. Okay. Where am I here again? It must be something with the way that I'm sharing the entire screen, and then I need to go. Okay, let's see. No. Deborah, do you still see see all the slides?
Yes.
Okay, so I'm going to try to do to start the slideshow now, again from my screen.
Okay.
And you tell me if it works.
Okay.
Okay. Does it work?
No.
Okay. All right. Is it working now?
No. You
No.
want to send it to me, and I can present for you.
Yes, I'll do that. Okay.
It's so strange because when we tested, it worked perfectly, right?
Yeah, but then there's something. Okay. So. I'm sorry for the. Oh, I can continue with the. If you see all the slides, it doesn't really. It's really big. I'm not sure it's going to be. I'm going to be able to share it to you. Send it to you. Did you receive it?
Not yet.
Okay, I'm going to try again.
No, I did. Now I did. Okay, so I will show your presentation, and you
Yes.
tell me when you want me to change the slides, okay?
Okay, perfect. Thank you, Deborah.
My pleasure.
Talk about stress.
Yeah, of course. What is life without stress?
Yeah. Okay. So.
Okay, we're ready.
Next. Yeah. Next. Yeah. Next. Yeah. Next. Okay. So now, if we look across species, we have a number. If we talk about early adversity, those have, of course, different characteristics depending on the species. In humans, we're talking about sexual or physical abuse, psychological abuse or neglect, poverty, low SES, also in migrations, and those can very well be sex dimorphic exposures, because in fact we know that women or men are differently exposed to, for instance, neglect or sexual physical abuse. In humans, in non-human primates, there's generally accepted that there's a transgenerational transmission of abusive behavior, abusive maternal behavior that could lead to early adverse environment. And in rodents, we have a number of different paradigms of early adversity, again, that can lead to sexual dimorphic consequences on the offspring. In particular, you can use maternal separation, maternal deprivation, limited bedding, and early weaning or unpredictable stress as models of early life stress in rodents. If we look at the effect of those early adversity situation on the development of the corticolimbic circuits, which we'll be talking about, in particular the medial prefrontal cortex, the amygdala, and the ventral hippocampus, there are a number of parameters that can be modified in the circuitry. It could be the developmental trajectory, either an acceleration of maturation or a delay in some instances. The modification in critical periods of plasticity, in the establishment of the proper balance between the excitation and inhibition, and also, of course, morphological changes that can occur in a number of different cell population. So next one. So we have been interested in looking at the effect of early adversity, because in using a model where we can observe fragmented and disorganized maternal care, some investigators also have found rough and maternal handling and really true abuse, which we haven't actually documented in our model. So I just wanted to make the distinction, because there are some different effects of this model. So we are interested in the development of the emotional dysfunctions, and also, but I won't have time to talk to you about that, in the development of altered circadian rhythms in those offspring that are exposed to early adversity. Next. So how do we generate early life stress in rodents? As I mentioned already, you can modify the amount of contact that the mother has with the infant using either the maternal deprivation or the maternal separation paradigm that have been well used and well documented in many different labs. And we've chose to use the limited bedding and nesting paradigm, which in fact, we don't intervene so much in the cage of the animal, but the mother is raising her babies on a platform here, and so she has really very limited access to bedding to make a proper nest. So this really results in a high arousal from the mother, I would say, and really a much more fragmented behavior. The mother becomes hypervigilant. And as I said, in our hands, we haven't really noticed a lot of abuse per se from the mother, but certainly a disrupted behavior. Next. Yeah. Just to show you that we've done some evaluation of the HP axis, very rudimentary, if you want. We've looked at basal and stress-induced HP activation. And at 2 different ages, day 10 and day 20, I forgot to mention that our limited bedding paradigm starts on postnatal day 1 and ends on postnatal day 10. After postnatal day 10, all the litters are put back on the normal bedding conditions. So it's really, our manipulation happens during the first 10 days of life. So if we look at the response to stress during on day 10 and that on day 20, what we notice, and in fact we were a bit surprised about that, is that we don't see much differences either in ACTH or corticosteroid under baseline conditions. But rather than have an exaggerated response, which I would find it a bit surprising in postnatal day 10 animals, because we're still in the stress hyporesponsive period, we actually see a reduction in the response, in the ability of the HP axis to respond to stress. And the stress we used here was immobility stress. On day 20, we see a small effect on ACTH here and no effect on corticosteroid. So really, it seems like much to what has been documented maybe in PTSD, except that in adult animals, we have a functional HP axis, but here we also have a blunted response, which might just signal that the animal ability to respond to stimulation is reduced. So next. In those animals, of course, what we observe and what was the primary behavioral endpoint is an increased freezing responses to conditioned fear responses. And in fact, these are adult data, and it's only in males that we found an increase in freezing behavior, and we didn't see the effect in females. Next. So if we look more closely at the corticolimbic system, we're talking about the prefrontal cortex here, prefrontal cortex here, the basolateral amygdala, and the hippocampal formation, in particular, the connections with the ventral hippocampus, which are important. I don't know, do you see my mouse or you don't see my mouse, of course, right? Yeah, okay. So in our studies, we've also looked at differentially between the prelimbic and infralimbic prefrontal cortex, because typically the prelimbic area is important for the acquisition of fear, and typically the infralimbic area is more implicated in the inhibitory fear extinction and fear inhibition, although both regions are actually implicated as well. At the level of the basolateral amygdala, we have often separated the anterior from the posterior amygdala because those regions might be quite different functionally. Next. So the question we've asked really is what effect has this early life stress on the basolateral amygdala, and does it have the same effect as chronic stress in adults, which is known to, in fact, enlarge the amygdala, increase spine density in adult animals? And to our surprise, what we found is, in fact, already by day 10, we have an increase in spine density in the basolateral amygdala of animals that are subjected to limited bedding compared to the normal bedding animals. And this is day 10, and this is day 20. So I'm sorry, because my slides are really small on my screen. So I'm going to try to actually point to the right thing on the images. So next. I have to say that when we did the quantification of the morphological parameters of those basolateral amygdala neurons, and this is shown either on day 10 or on day 20 for spine number, dendritic length, and number of branch points. And what you can see is that already by day 10, we have we seem to have an increase in spine number that becomes significant on day 20. On day 20 also, and I want to remind you that on day 20, they're not on a limited bedding condition again. I mean, the limited bedding condition stops on day 10. So this is a lasting effect here that we notice in dendritic length and as well as the number of branch points. And I want to specifically say that this is something that we have observed in male rats, but not in females. Next. So what are the consequences or the functional consequences of those morphological changes? We did a number of electrophysiological studies, and without going into much detail, what we observed is a trend towards an increased action potential of those BLA neurons in male rats. Nothing happens in females again. And so that's when we did the patch clamp studies. Then we wanted to look at synaptic plasticity. And for that, we used a classical long-term potentiation protocol. And much to our surprise, the only effect that we noticed really between the normal bedding and limited bedding were always in the right amygdala. Nothing happened in the left amygdala, and it was mostly for males and not for females. The females are the bottom panels here, the C and D. So not only were we able to demonstrate that we have an effect in males of those morphological changes to increase a long-term potentiation, synaptic plasticity in the males, but it was also differentiated by hemisphere. And as you know, maybe the literature on the hemispheric specialization is actually well developed for humans, as we know that the right amygdala is usually fairly more responsive than the left amygdala. However, in the rodents, the literature on lateralization is really not that well developed, and you don't find a lot on that. And so we were very surprised to see it, which demonstrates that now, in addition to have males and females, we have to have left and right as well when we do our measurements. Next. So that was for the principal neurons in the basolateral amygdala. And we said, okay, so if we have changes in synaptic plasticity and excitability of those cells, does that mean that we have somewhat of an impairment in the inhibitory functions of the interneurons that are very critical for all the circuitry that modulate fear acquisition and extinction? So we just run a little study looking at activation of paravalbumin cells, the primary inhibitory interneurons in the basolateral amygdala. And again, we did left and right because we wanted to make sure that we have the right measurement, and in males and females. And what you can see in the open bars is basal responses to control. And in the hatch bar is the response to fear. In males and females, actually the hatch bar is the limited bedding, and the full color is, the first set of bars, I'm sorry, is the control. The second set is the fear. And if we concentrate on what happens in the right amygdala, what we see is that after fear, we have a reduction in the FOS activity in inhibitory interneurons, paravalbumin positive interneurons. So that suggested to us that maybe the increase in excitability of those BLA cells is due to the fact that those interneurons are somewhat less active at that time. Next slide. So I just want to quickly go over something that is very interesting for the activation of inhibitory interneuron, parvalbumin inhibitory neurons, and those are the perineural nets. Perineural nets are usually molecules that are derived from the extracellular matrix, and they are composed on a very, very complex structure of hyaluronan backbone that is connected by those CSPGs, which are proteoglycans. That is hard for me to actually pronunciate, and by link of several proteins. So it's very large assemblies that actually develop around most parvalbumin positive inhibitory interneurons. And in fact, what is very interesting about those perineural nets is that they mark the closure of critical periods of plasticity. So the onset of periods of plasticity is marked by the development of parvalbumin immunoreactivity and the development of parvalbumin interneurons. And once those interneurons become surrounded or encapsulated by those nets, then those nets actually sort of stabilize the synaptic inputs to those interneurons, and it reduces the plasticity of those interneurons. So the question for us was, okay, it seems like we have a reduction or at least a modification in the inhibitory tone at the level of the BLA. So does that mean that, in fact, the early adversity also modifies the occurrence of those perineural nets? So next slide. And in fact, we did a study. We used immunocytochemistry to look at those perineural nets. And I forgot to show you on the previous, but I can show you on this one. The perineural nets are those molecules that are green around the red cells that mark the parvalbumin cells. And so they're really around the cell. So if we try to quantify the number of cells that express, the number of PV positive cells that express PNN around them, we can see that in the basolateral amygdala, we have more in males than in females. And in fact, we have more in the limited bedding condition, which is the black bar that you see here. Again, nothing happens in females. So what are the implications of that? Does that mean that early adversity could cause a premature closure of critical periods in the basolateral amygdala, and therefore sort of freeze the phenotypic characteristics of interneurons that are in a different state that they would be if they would have been allowed to mature adequately? So this is something that we are pursuing a little bit right now. Next. So the way that we see this in terms of the local circuit and organization in the basolateral amygdala is that in males subjected to limited bedding or early adversity, what we think is happening is that we have an hyperexcitability of those principal neurons, glutamatergic neurons in the basolateral amygdala. That is due to the fact that we have a reduction in the activation of interneurons or inhibitory interneurons that provide inhibition on those principal neurons. So we have an increased right BLA activity. Now in females, and I haven't shown you everything because for the sake of time, but we don't see much happening in terms of PNN around parvalbumin neuron. We don't have changes in the activation of PV interneurons. And in fact, what we see here is that we have changes in glutamatergic NMDA receptors and no change in AMPA receptors, and we even have a reduction in the action potential in females, meaning that already very early on, we can see a sexual dimorphism in the way that the system responds at the circuit level. So next slide. We wanted to know if early life stress influenced connectivity in the cortical limbic circuits. So we did some fMRI resting state studies in young pups and adults. Next. I'm going to go fairly quickly because I just want to mention that in humans, in fact, there are many studies that have shown that it seems that there's a more adult-like connectivity between the amygdala and the prefrontal cortex that has been seen in infants that have been exposed to early adversity. And this is a study by Nim Tottenham looking at children raised in orphanages, where you can see that in the normal maturation is that you can see a negative, more adult-like connectivity happening later on in normal infants. And in fact, this happens prematurely in infants that have been exposed to previously in orphanages. So we wanted to see if we had similar changes in our preclinical model. Next. And I'm going to go fairly quickly because I don't know how I'm doing in terms of time. But basically what we noticed and we observed a reduced connectivity between the amygdala and the prefrontal cortex that was already observed in males after early adversity and continued also in adulthood. So next. And again, we had different seeds that we looked into the interior BLA or the posterior BLA, and again, left and right. And you can see here that with the prefrontal cortex, for instance, the right BLA, the interior BLA, was consistently a reduced connectivity between the anterior right BLA and the prefrontal cortex, the PL or IL regions. Next. So in summary, the limited bedding conditions that we use as our model of early life stress produces significant morphological and functional changes in the amygdala, in particular the basolateral amygdala, with enhanced spine density, excitability of principal neurons, and increased plasticity in juveniles. These changes are seen mostly in males and more prominent in the right amygdala. We also have observed possible changes in inhibitory interneurons and also in the closure and the premature closure of critical windows of plasticity in the basolateral amygdala. I have to mention that we also looked at the prefrontal cortex and are continuing those studies, and the situation is the opposite in the prefrontal cortex. Early life stress modified connectivity between the BLA and other brain regions, in particular the prefrontal cortex, and in particular with the right BLA. What are the implications for development of fear and anxiety as a function of sex? Next. And an ongoing questions is really, how does early adversity program the increased fear and anxiety differently in males and females? Do we have early organizational effects of gonadal steroids that are implicated, and at which level? How does early adversity affect hemispheric specialization and laterality? We have some example of that in our rodent model. There is also example in the literature on humans that the right amygdala is mostly affected. And again, another line of interest for us is, how do you transmit the signal of fragmentation of maternal care into the establishment of circadian rhythmicity in neonates? Because we know that the establishment of rhythms is important for a number of physiological functions, and then how is this behavioral translated into a biological system in the infants. Next. So now I wanted to thank my team, and it's an all-women team, and that have been working on this project, and I'm going to leave you with that. Thank you very much. That's it.
Thank you very much for this wonderful presentation. And now we have Dr. Michael Kober's presentation, and afterwards we'll have a general discussion, okay? Thank you so much.
Thank you, Dominique. Thank you, Andrea, for the great talks. I sure have many questions for you both, but we will be able to talk a little bit later on, because, well, now it's a great pleasure to have here today Dr. Michael Kober from the Therapeutics of the University of British Columbia in Canada. Dr. Kober is an affiliated scholar at the Human Early Learning Partnership, or HELP, which is a very nice acronym, by the way, which is a collaborative interdisciplinary research network based at the School of Population and Public Health at the University of British Columbia. Michael is also kindly collaborating with us in our project about biosocial, biopsychosocial epigenetics and its inheritance, especially paternal inheritance. That's why I was very interested in Dominique's talk. What has been actually essential for us to put the project together. So Michael, thank you very much for joining us, and the screen is yours, and Gabriela will share your slides. Thank you.
Okay. Well, wonderful. Thank you so much, Taisa. It's a real pleasure and an honor to be with you these few hours today. I look forward. Very much enjoyed the presentation so far, and look forward to discussing our work with you. So, are you going to show my slides, or hello?
Okay, Gabi.
So maybe while Gabi gets ready, so what I'd like to do today is really highlight for you our work at a very high level of epigenetic embedding of early life experiences, and really focusing on a paradigm from society to cell. So I will show you, really focus on two things. First of all, I'm gonna discuss with you epigenetic associations of a number of different situations, really focused on early life. And secondly, I'm going to talk to you about a new concept that we're very excited, fairly new concept that we're very excited about, which is epigenetic age and how that might relate to children's health. So there we go. So I already went 2 slides in. So if you go to the next one. Yeah, so really, and focusing, and hopefully with this take-home message that at the epigenetics really is at the interface of nature and nurture. So if we go to the next slide, please. We already heard a bit about epigenetics this morning, but there are quite a few different definitions of what it is. Here is perhaps the most popular one. If you go to the next slide, our definition really is much more about the social processes and really in how early life experiences influence the ways our genes are expressed. So our paradigm is essentially that there are experiences that get under the skin to affect biological pathways, such as some of the ones we heard earlier, and then drill all the way down into the nucleus to affect the expression of genes, and in some cases doing so in a permanent and persistent fashion. Of course, this is not a one-way road, so if you go to the next slide, because we can also easily imagine that that go in the other way. And many different players are part of what we call epigenetic processes. So if you go to the next slide, this is from a figure highlighted from a review from my former postdoc, Maria Aristizabal, who really beautifully highlighted the complex landscape of all these different epigenetic marks and mechanisms. And really, every single one of them has a fantastically interesting story to tell. But for today's presentation, we're going to exclusively focus on one of them, and that's at the very upper left, DNA modification, and in particular, the methylation on cytosines, and usually in the context of CpG dinucleotide, but not always. And this is really the mark that especially human populations, I would think at this point, 99% of all studies have been done at. We're slowly seeing other marks emerging in human populations, but for the students out there, if you look at all these other players that make up the epigenome, and there's lots of work to be done. So please, go ahead and think about all the different epigenetic players that could contribute to what we're talking about beyond just DNA methylation. So I give a lot of talks to public, so I often, because this can be kind of overwhelming, I try to sort of put a different spin on what epigenetics is. So if you go to the next slide, please. What epigenetics is, if you go next slide, it's really to some extent very simple. If you think of light bulbs, you can have a light bulb that's either totally on or totally off. Or if you live in a fancy house, you might have a dimmer switch, and you can put your light bulb anywhere in between. And so to me, what really is happening is that when we look at our genes, those are the light bulbs, and the epigenome is the dimmer switch. So if you go to the next slide. Again, yeah, and so if we, for example, look at, Taisa's favorite gene, I don't know what it might be. In this case, it's totally on, it's fully expressed, light bulb is on. And now we start to slowly methylate on these CpGs, the promoter. So if we go to the next slide, you see there's a little methyl group coming in at the top. Next slide. One more, and next slide. So now we have fully methylated Taisa's favorite gene. We push that dimmer, that epigenome all the way down, and we turned the light bulb off. So that's all you need to know about epigenetics, quote unquote. So this happens during development all the time. It happens in all of our bodies, in all of our cells. And in fact, some of what makes a cell, given cell, is actually basically embedded in these epigenetic patterns. But I think what really has become one of the most exciting areas in biology, and one of the most interdisciplinary areas in biology, is the fact that epigenetics actually can also be to some extent associated with different environments. And so if you go to the next slide, there is, for example, really compelling work out there that the nutrition and the lifestyle of the mother can affect the epigenome of the child, usually sort of based on sort of natural situations, such as here the Dutch famine, where, for example, was documented that even 60 years later, there were epigenetic marks, some of these dimmer switches that associated with whether the mom was undernourished or malnourished during pregnancy or not. Many of other studies, including those focused on maternal smoking and the like, have documented now similar effects. If you go to the next slide. Perhaps even more tantalizing for the public is seminal work by our previous speaker's colleague, Michael Meany, has shown that the quality of the maternal care is often associated with the epigenome of the child. In some cases, going all the way to linking early life abuse and suicide through epigenetic marks. If you go to the next slide, please. And then, of course, there is also really interesting work that if you look at identical twins who, by and large, have the same genome, but don't always do the same. So if you look at cutie on the right here and screaming baby on the left, both of those have especially early in life, have almost identical epigenomes, but as they go through life, their epigenomes diverge and become more discordant, and especially if they happen to have been separated early in life. And in some cases, This can then also be in turn associated with different diseases. So, for example, things like even mental health and cardiovascular health in identical twins has been associated with differences in their epigenome. So collectively, if you look at the next slide, these and many other exciting data then leads us to look at the epigenetics in child health, as I said, as the interface of nature and nurture, where really our genetics, as you can see in the left by the DNA here, is the nature, and the nurture is sort of that green, fertile family environment. Together, we try to sort of put these three domains together to understand how we can best provide them healthy starts to our children and to prevent diseases that are rooted in early childhood. And if you go to the next slide, please. We do this, of course, in a very developmental context, because what has become clear that there are certain periods, sensitive periods for particular exposures, and often a lot of focus, of course, has been on the first few years of life and going all the way back to conception. When we need to realize, as my former postdoc Meg Jones so beautifully illustrated here in this figure from Annual Reviews of Psychology, that while our genome stays the same more or less throughout life, what changes is really our epigenome. And for example, if we have a particular exposure, like an early childhood adversity that we heard earlier today, in this sensitive period, it might alter the trajectory of the epigenome, as indicated by that red line, to result in a higher risk for health problems down the road. If, however, the same exposure might happen outside of this sensitive period, it might not alter much the trajectory of the epigenome, as indicated by the blue line, and thus might not lead to an increased risk for adverse health outcomes. My good friend and former mentor and hero, the late Clyde Hertzman, really, even before the emergence of epigenetics, put all of this beautifully in a conceptual paper where he talked about this biological embedding of these early life experiences and how they might have profound effects of health in adulthood. And of course, many of you will be familiar with the developmental origins of health and disease framework that is very similar to this. So if you go to the next slide. So we've been trying to put, with a suite of different studies, these concepts to test using real-life populations. And particularly, we have been focusing on a few areas related to early life environments, specifically early life socioeconomic status. So does it matter whether you grow up in a little dwelling like you see on the left, or a mansion like on the right, or early life parental stress? Does it matter if you grow up in a nuclear family with doggy woof woof, or in a family where the parents constantly yell at each other? And ultimately, how does that, of course, affect childhood temperament, behavior, and trajectories? And we've been doing this work really standing on the shoulders of giants, with a large number of wonderful collaborators that you can see, the main ones of those on the blue box at the bottom there. And so I want to just highlight a few examples that we over the years have been working on and documenting, showing that I believe indeed early life experiences get under the skin in part through these epigenetic mechanisms, the circuitry between those dimmer switches and the light bulb. So if you go to the next slide. In one of our earliest pieces, together with Greg Miller and Edith Chen, we actually documented that if you look at a cohort of adults, healthy adults, that is stratified by socioeconomic status in early childhood, so for the first 5 years, but mixed for socioeconomic status in adulthood, you can see a few distinct patterns very consistent with our overall hypothesis. So first of all, if we look at the light bulbs at the left there, we can see that in the white blood cells of these people, there are differences in these light bulbs. These are not random differences. So these genes that we find to be differentially expressed are actually not just random genes, but in fact, if you do bioinformatic analysis, it turns out that there are genes that are collectively creating a pro-inflammatory phenotype. So you see, for example, NF-kappa B and glucocorticoid receptor regulated genes being enriched in this gene set. Importantly, when we then put this to test, we find that in the white blood cells that are derived from folks with low socioeconomic status, we actually see an overreaction of the innate immune system, as you can see on the top right. These are TLRs, or toll-like receptor pathways that we stimulate here and test. And in each of the cases that we tested, the cells taken from folks who grew up in low socioeconomic status had a higher activity of their of this innate immune system, very consistent with what the gene expression profiles predicted. Further, if you look at cortisol, at the bottom right here, we can also find that collectively folks who grew up in lower socioeconomic status have a higher cortisol output throughout the day. In work that I don't have time to show, we then could actually show that to some extent there is a buffering on the resilience of these patterns that can be associated with maternal warmth early in life. If you go to the next slide, please. When we then look at the same people, we can also show, we also found that DNA methylation, so these dimmer switches, are also correlated with early life socioeconomic status, as indicated by these skewed p-value distributions. Interesting, if you look in this particular cohort, we only find that folks who grew up in early life SES, we find an association with early life SES, but not with current socioeconomic status at their age of 30 or 40. So collectively, these data, early data, suggest that early life poverty, socioeconomic status gets under the skin by associating with changes in the epigenome, persistent changes in the epigenome, by basically sculpting gene expression patterns that are associated with pro-inflammatory phenotypes, and in fact, with changes in immune system response. Now, of course, these were early days, and in our world, you always want to see to what extent you can replicate these data and perhaps become more sophisticated. As you probably realized and already heard earlier, socioeconomic status is a very, very difficult and very complex construct, and so we have been looking at other situations to test, to ask whether we can replicate these findings. So if you go to the next slide. So more recently, together with Tom Boyce at Nikki Bush at the University of California at San Francisco, we have looked in an adolescent cohort, so slightly younger than the previous work, to the extent that we can actually also find associations with components that make up socioeconomic status. And as you can see here, if you just want to focus on the right, we find a number of dimmer switches associated with things like again, income pair dependent, but also parental education, and even family adversity. And so some of these are overlapping, but there is also a number of unique associations. But collectively, I think it illustrates that, yes, even in a very different cohort, at a different age and in a different tissue, these are now buccal cells, we can find associations between early life components and DNA methylation marks later in life. Further, if you go to the next slide, we have also taken this into low and middle income countries. And so this is the work we've been doing with Tom McDade and Chris Kusawa on the Cebu longitudinal study of nutrition and health in the Philippines. And again, without going into too much details, you can see in the Venn diagrams on the right that we find a number of associations between early life environments, again, education, income, and the like, in this adolescents or young adults in the Philippines. And to give you one example, on the left here, this is what's called a volcano plot. That's a statistical way of showing the data, where you can see what you look for, just like in a volcano. You look for dots that go further away from the middle. So the green and the red dots, those are biological effect sizes. And then you look at dots that go very high up, because the higher up you go, the more statistically rigorous you get. And so you can see, for example, that here we find almost 2,500 of these dimmer switches that are associate whether you grow up in low versus high socioeconomic status in this cohort in the Philippines. So collectively suggesting that, yes, there is certainly associations of these early life socioeconomic status and the like across different cultures, across different tissues, and across different ages. And if you go to the next slide, please. There are many more examples that we have on this. So, for example, we have shown, together with Marilyn Essex and others, that we find associations of family stress with DNA methylation dimmer switches in adolescents, together with Megan Gunner, in a similar paradigm that we heard in the previous study from Nim Tottenham with these, the second study. It's about where we looked at children that were adopted from Eastern European orphanages into the United States. And again, we find a signature on DNA methylation that can be traced back to this early childhood adversity that these children experience for the first 2 years in life in these orphanages in Eastern Europe. And another sort of striking example is at the bottom. Work with Andrea Roberts at Harvard. We found that even in human sperm, we can find that there is a signature of childhood abuse, sexual abuse that these men had been unfortunately experiencing early in life, that we can see many, many years later to be associated with these dimmer patterns in the sperm. So I think collectively making a strong argument that diverse social and environmental exposures and experiences can indeed get under the skin to affect these dimmer switches and potentially health. So if you go to the next slide, please, Tessa. So what I've shown you so far is really all associations of these epigenetic associations, and they're really interesting because they can identify, particular genes or even particular pathways like the immune pathway that are associated with various environments. They are also a little bit challenging in to some degree, and I touched upon this because just by the nature of the epigenome, it differs between different cell types. So, for example, blood and brain and buccal cells all have distinct dimmer switch patterns that occur, that sort of develop naturally as our bodies mature. And so the question becomes to what extent some of these signatures are really reporting across tissues versus just for one tissue. And this is particularly, of course, relevant when we think of making associations with behavioral phenotypes that happen in the brain, when in reality, we don't really have access to living brain, from brain samples from living people. And we can talk about this later. And of course, these measures become rapidly more and more complex. So when I started this work 11 years ago, we measured, I think, 1,500 dimmer switches in parallel. Now we're measuring 1 million in parallel. And so the statistical difficulty of these things becomes increasingly complex. But nonetheless, it's fascinating, very insightful. But what has happened over the last few years is that the community then has also asked, well, are there other ways by which we can exploit the information content of the epigenome? And Steve Howard in particular realized that, as I mentioned earlier, these marks change during development, they change during aging. And so the idea then became, can we predict aging or compute aging based on these epigenetic marks? And that's what Steve Howard did, in fact. And he did it in a way that is the same across almost all tissues, just by the way he constructed his algorithm. But basically, what he then came up with is this idea of epigenetic aging, of epigenetic clock. And the statistical difficulty of this is very, very simple, because basically you only compute one number. That is the difference between your predicted epigenetic age and your actual chronological age. And so to make this a little bit more clear in the next slide, please. Again, my former postdoc, Meg Jones, put this, I think, very succinctly in that, we look at, say, if you compare chronological age with predicted epigenetic age, the vast majority of us, just by the nature of how these algorithms were designed, are in these concordance zones. In other words, our chronological age might be more or less the same as our epigenetic age. But then there are some folks who are epigenetically old, and then some folks who are epigenetically young. So, for example, epigenetically young would be somebody whose chronological age is 40, but their predicted epigenetic age would be more like, say, 25. And epigenetically old would be the same 40-year-old person, but their epigenetic age would be 50. And then that in turn is associated with different health outcomes, with, of course, the epigenetically older having poorer health that at earlier ages compared to the concordant folks, and especially the epigenetically young folks. This has been now shown many times. There are all kinds of conditions that are associated, particularly with being epigenetically old. Things from being obese to having neurodegenerative diseases to PTSD, all the kind of things that you would expect. There are a few things that are associated with being epigenetically old, and this audience will appreciate that one of them that has been repeatedly shown to be associated with being epigenetically young is being of Latin origin. So that's good. Now, we have now then put this to test in many ways. And if you go to the next slide, coming back to our sort of flagship study, that of being, what's the effect of early life socioeconomic status, in the same paradigm that I showed to you in our first data slide. What you can see here is, if we now, rather than looking at epigenetic pathways, we look at epigenetic age. What we find is, and perhaps not entirely unsurprisingly, is that folks who grew up in low socioeconomic status, irrespective of their current socioeconomic status, tend to be epigenetically older than folks who grew up in high socioeconomic status. And just to put this in perspective, in lots of epidemiological studies, has been shown that being epigenetically older in turn is also associated with overall increased mortality rates in populations. So again, showing that early life gets under the skin to affect and associate with differences in epigenetic age many years later. Now, as we were doing this work, we also realized that these epigenetic clocks, perhaps not entirely surprising, they were constructed, and there are many of those epigenetic clocks now, they were constructed almost exclusively for adults. A lot of our work is really interested and focused on children. So if we go to the next slide. My former PhD student, Lisa McEwen, then decided to team up with Steve Horvath and create what we think is a much improved epigenetic clock for the pediatric age range, and specific for buccal cells. And so what Lisa did, she basically used machine learning and divided our data, in-house data, into a discovery, into a training, and into a test set. And what you can see here is a very nice correlation between predicted epigenetic age, buccal epigenetic age, and chronological age across the pediatric age range, and the replication thereof in our test data set. Now, when Lisa then starts to ask, well, what is the epigenetic age, these deviations of these children that you see that are not right on the diagonal line there, what is it associated with? And so if we go to the next slide, we can see, for example, that it is associated to some extent with gestational age. So changes gestational age associated with differences in these children's epigenetic age, but perhaps more excitingly for us, it's also associated with, in this case, at the bottom, autism spectrum disorder, using publicly available data where children that in red that have autism spectrum disorder are epigenetically a bit older by this measure than control children. So this was very early data for the paper. Just give you a flavor of things to come. But if you go to the next slide, we now have a large building a large network to put this, what we call the pediatric buccal epigenetic clock, into context. We got some really nice funding to look at, what are the intrinsic factors, so sex, genetic background, ethnicity, and such, that interact with the clock. What are the physical and social environments, the things like, during pregnancy, smoking, drinking, the socioeconomic status and such, and that associate with the clock, and also how are interventions potentially slowing or accelerating the pediatric clock. And so collectively, we try to understand basically whether this clock is at the interface between these intrinsic and extrinsic factors and health and developmental outcomes at the bottom. And I invite all of you to become a partner in this journey. We are always looking for new cohorts. Again, it's buccal cells and it's children. So if you have any such cohorts and are interested in partnering with us, please get in touch. My email is in the orange box there. So if you go to the next slide, Kaiser, please. So I hope what I convey to you is really using some high-level examples of our work, that the epigenome, these dimmer switches, and the potentially the light bulbs, the gene expression, are at the nexus between early life experiences, whether these are social experiences or physical experiences, and children's phenotypes. I think a key function here is in part on the immune system, but I just want to highlight that as we look at this, we also need to be aware that these things, of course, are different between tissues and that they change during development. And didn't have time to talk to you about it today, but there is also a strong component of genetic influences that is emerging to be highly associated with epigenetic patterns. And so if you go to the next slide, please. What's really interesting, of course, if we think of this also in the context of the pandemic, which I'm sure you're all too aware of, but together with some colleagues, we have put out a year ago already a piece that really talks about that there could be long-term biological effects, because many of the things that I just showed you, whether it's poverty, whether it's stress in the family, whether it's nutritional shortcomings, all of those, of course, are generally got worse, especially even here in Canada during the pandemic. And so you can only imagine how some of these things might then associate in turn with brain development and epigenetics and the like that we have been discussing here today. But just in my last slide, my next slide, please. I leave you with this. Is this nature versus nurture, or is it nature and nurture? And I believe that balance is very much an interplay between the two. So thank you very much, Kaiser, for the invitation. Did I get kicked out? No.
Okay. Can you hear us? Okay. Thank you very much for this very nice talk. Thank you for sharing with us very brand new information. So it's very exciting actually. I'm not sure if we already have any questions from the audience, but I think we can all be here in the panel. Please, Lincoln, can you put all here so we can have a brief discussion? Dominic, Andrea, Deborah, Claudia, please.
I have a question. I have some questions for Michael, if I may.
Sure, please.
Yeah. Well, thank you. Thank you, Andrea. Thank you, Michael, for this fantastic talk. Michael, I have two questions for you.
Sure.
And one is, when you talk about epigenetic aging, do you consider that to be sort of a recapitulation or an integration of significant events that happen to an individual? Like, is that like a composite score of what we could call overall hits?
Yeah, it's a great question, Claire. I think it's still too, so I think in adults, certainly, we see all these associations of, a lot of different things that can happen. And in many cases, each of them, associating with changes in epigenetic age. And so I guess you could argue that maybe that epigenetic age is a sort of a readout, a score of things that have happened to us. And often, the bad things actually, as you perhaps, I think I touched upon the bad, quote unquote, bad things, like, you talked about PTSD and things, associate with acceleration in epigenetic age. I don't know to what extent, people actually have kind of created a sort of, say, like a large variable. Like all of these studies look at one or two variables. I'd be very curious, and now that you bring it up, I'm actually have to look more closely to what extent some of them might act together to accelerate or decelerate epigenetic age, or whether they're all sort of individually associating, if that was your question.
Yeah.
In children, we basically know almost nothing. So,
yeah, because if you imagine like you have a blueprint of normal aging,
yeah.
And that you superimpose like all those different factors that can affect it, then maybe that will be the composite score that you get when you look at this epigenetic aging or differences in chronological versus epigenetic aging, right?
Yeah, no, that's a great point. It's sort of like, is there sort of an allostatic load of epigenetic aging, so to speak?
Yeah, exactly.
But that's a great, and for children, I mean, yes, certainly in adults, it's quote unquote bad for you if you're epigenetically older. Well, for children, we actually don't know. Like, nothing is actually known, and so we're actually, using now also like the kind of stuff you were talking about, like rodent models, because you can actually do the epigenetic clock now in mice and in rats as well, to sort of look, to what extent really in childhood is it actually quote unquote good or bad for you if you're a little bit, more advanced epigenetically, say at age 2.
Yeah, but I mean, and the definition of good and bad is really hard, because let's take the example of early adversity and the fear responses, for instance. So children learn very quickly to recognize the signals, and they sort of you get probably a faster maturation of the system, but ultimately it's not a good thing. So it's good in some aspects for adaptation to a situation, but later on it turns out that it's really bad, right?
Right. And just highlighting, I think, as we all agree, that there is really, you need to look at this all in a very developmental context.
Yeah, and also different systems.
Yeah, for sure, yes.
Okay, thank you. Andrea has a question for Dominic, is it?
Yes, yes. Thank you for both Michael and Dominic's amazing presentation, and I think we were able to add up to all the presentations, a little piece here, a little piece there, and. One thing that, Dominic, I'm not sure if I missed in your slides, but we know that depending of the early life event, the type of early life event, it does have more effect on each sex, right? Males being more susceptible to one kind of event, and females being more susceptible to other events. But what caught my attention in your slides, if I'm correct, is that you were able to show the sex effects on frontal limbic connectivity, right? On males having, correct me if I'm wrong, increased frontal limbic connectivity. Is that correct?
It was reduced connectivity.
Okay.
And in fact, those studies, we only did them in males at that time. Since then, we have repeated them in females as well. And so I'm anxiously waiting for those data. We're still analyzing them.
Okay, yeah. No, that's amazing, because we did at a similar evaluation with our DTI data, and we were able to see actually the reduced frontal limbic, actually cingulate and prefrontal cortex connectivity, but only in males, on which mothers were exposed to early life experiences. So I think we end up complementing our data, and it's interesting to discuss to which point the sex-specific effects they have on the development on fetal stages, right? Why is the male brain more susceptible than the female brain?
That's $1 million question, I think.
Yes, I know.
and I mean, one thing that is, I mean, in many of the models of early life adversity, at least in rodents, the males seem to be the most susceptible, okay? However, this is something that is so hard to reconcile with the human literature, because females seem to be a lot more affected ultimately in humans, So I think there are just probably major differences there that we haven't really grasped yet. For many preclinical models, it's clear that the vulnerability lies in males. What makes a female more resilient is really something that is fairly unknown at this moment. Whether it's the chromosomal makeup or the early effect of organizational steroids, this is something that we are looking into, because certainly, I mean, and already from our data, looking just at neuronal excitability and a number of NMDA receptors, AMPA receptors, etc., the system seems to be very different from the start. And so, yeah, and they seem to be reacting very differently from the start, very early on. So I can't answer this question.
And just a very quick point, Andrea, actually the epigenetic clock, the epigenetic age also tends to be different between females and males. I didn't have a chance to talk about it, but generally females tend to be younger, even though, it's not a question of the bias in the training set. So for whatever it's worth.
Yeah, there are many answers, and we are not even seeing that. We are just seeing the top of the iceberg, right?
Yeah, but it's really interesting because it could give us an idea of resilience, right?
Right.
And to know.
Too much to think. Deborah has some questions as well. Your microphone, Deborah.
First of all, thank you so much for this wonderful talks, and it really amazes me how close we humans are from rats, and how nice these animal models are, to how useful they are to answer several questions which are still very hard to answer in human beings. Our data are very similar in the sense that females are more resilient than males, not only in early life or infancy, childhood stress, but also in adolescence. We show that females are more resilient to some of our models, which is really interesting. And it makes me think that maybe women are more vulnerable because they are more exposed. So it's almost like we are building some vulnerability based on excessive exposure, or there is some societal and cultural exposure that makes women more vulnerable. Because biologically speaking, it doesn't seem to be the case. Well, this is one thing that I think that we have to really dig in and try to understand why animal models do not resonate what happens to humans. But my question for Dominique, it's more specific about the age effect. It seems to me that stressors that are imposed to pups until postnatal 9 or 10 are very effective in increasing the vulnerability for psychiatric psychiatric disorders or models of psychiatric disorders. After that, the animals seem to be resilient or more adult-like. They are not so vulnerable. And I wanted to listen to you to hear if you had reached a similar conclusion, and if you disagree with me, how would you explain this sensitive period ending at postnatal day 9, postnatal day 10? Because I was reading many models, Regina Sullivan's model, Kali Barams is the model you use, and everything seems to happen until postnatal day 10. Even the maternal deprivation, we see that maternal deprivation on day 11, the animals are much more resilient and they don't develop, anxiety-like behaviors. But before that, they are very, very vulnerable. So I would like to hear your thoughts.
Wow, that's, I think we should be talking about 3 hours about that. I agree with you. And honestly, okay, about the stress hyper-responsive period very early on. And people saying, okay, well, this during this paradigm, as you say, this window where it's really efficient, then, you have worked on that. I have worked on that a little bit also. We don't see a lot of, classical HPA axis activation, right? If I remember correctly, it's only after maternal deprivation for 24 hours that you see an increase in CORT in those animals, right? And we have not seen, and I've shown you that I was really surprised, but we have a reduction in CORT actually rather than an increase. Now, Regina Sullivan has a set of very interesting data looking at attachment and fear and the buffering effect of the mother. Something seems to be happening here around day 10, where the animal can actually start to recognize an aversive event. When the animal is conditioned to the aversiveness of the situation in the presence of the mother, despite the fact that the animal can recognize this aversive event, it will still consider that as not an aversive event, right? The mother is there to buffer. So I'm just wondering that we have a window, a very sensitive window. And when in this window, we happen to have an abusive mother or some kind of neglect or some kind of adverse environment that is superimposed to this window, then that is where, the system gets into a mode that is either an accelerated maturation in the case of fear processing, for instance, or possibly delayed maturation in the case of biological events that are not so important at the time for the adaptation, right? So it's like it's a trade-off, and so dependent on what happens on this stage on day 10, if you want. This is the sort of the window. I really don't know exactly, because I think, it's really hard to say. But I mean, CORT and norepinephrine release in the prefrontal cortex, something typically probably becomes online at that time that we don't necessarily know, because it would be fantastic to be able to do, for instance, functional MRI in young animals, especially around that time. Because then I think we would be able to see the very different structures all at once, because looking at one system or a few region is not enough. to set it up. I'm sorry, I don't have an answer for you, but I have just as many as you have.
It was a provocation. I just wanted to, to toss the question, because I think it's something that we need to start thinking about.
Yeah, definitely.
Yeah. It goes the same direction for humans also, because there is a window. There are some specific windows also in human development. So I just
Michael and Andrea, which one would there be about, like the first year of life or something, that you would think that would be critical?
I would say the first 300 days are the most critical period of the development, right? There are many processes that are speeding up at this time. So, and that's what we're also talking about. And males and females, they do have the depending on the type of the adverse effect, if it's a physical abuse or if it's a sexual abuse, it affects males and females in different ways, even though it happens at the same period of time, right?
Yeah, I agree, and certainly, from the epigenetics, one would sort of see the first, obviously, in utero, but then the first year or two. But I've actually become, and I'm not sure whether there's a bias just because we have been studying early life so much. We've been recently become very interested in puberty in adolescence, because if you think about it, there's, and maybe it's just because my son, I think, is going through puberty right now. So the changes that are going on in terms of, body, hormones, behavior, it's mind-boggling. And so, I'm starting to think whether there could be a second sort of area, window for plasticity that's a bit more pronounced than perhaps, at other times. And we just haven't studied it as much as the first, year or two. So I'm getting very fascinated by that.
I completely agree. Completely agree. Yeah, there are a couple of windows of vulnerability, and we are focusing here on the early years, right? Intrauterus and early years, and we are forgetting about puberty, right? Right, correct.
And it's many of those are actually associated with changes in sex steroids, right? We could then in a few years we'll look at the epigenetic changes happening around andropause or menopause, because we'll have we'll have this ability.
Okay, very nice discussion. We are coming to the end of our meeting, but I have, if you guys have just more 5 minutes for me, I have one question for Michael about the data on the autistic children. Did you guys look at boys and girls, or only boys?
No. So that's a great point. So this is a public data set that we downloaded from the public database, and we had very limited information other than autism, yes, no, and it was a small data set, and it was at a pretty strong spread of ages, as you could tell as well. So again, it would be very interesting, because of course there are differences between boys and girls in autism. And so as part of our new study, in fact, my next meeting is with somebody who has a really large autism cohort here. And we'll hopefully be able to look at this with a much more sophisticated and sex differentiated, way without losing power.
Can I have one more question? Just quick question.
Please, Dominique. Yes.
For Michael, the epigenetic aging you have based mostly on DNA methylation, right?
Yes, almost, entirely on DNA methylation. Yes.
So would you expect, or would you suspect that you would have different ages or chronological and epigenetic changes if you were to look at microRNAs, or if you were to look at HiDACs or things like that?
It's a great question. I honestly don't know whether anybody is doing it. I mean, Isabel Matsui or somebody that measured, these small RNAs would be well positioned in the animals. In humans, there's almost no study that I'm aware of that, has the kind of enough depth to look at things other than DNA methylation. We compared it already with telomere length because, of course, telomeres, very much kind of the same idea, and there is very little correlation either in adults nor in children between telomere aging and epigenetic aging. So I think they're telling us quite different things. It's really interesting to what extent, it correlates with some of these other epigenetic measures or chromatin measures, but also with all kinds of other clocks. I mean, I talked to my microbiome colleague the other day. There's a microbiome clock now. There are, of course, brain aging clocks. And so there's a lot of opportunity to cross-reference and see to what extent they correlate versus tell you different things. Great. Lots of work to do.
Dominic, we are planning to do this in our project. So it's kind of
great.
Going to start, I hope, by the end of the year, after all this pandemic thing is over. I hope here in Brazil. So I hope we can come back here and discuss this new point of the epigenetics and epigenetic age, especially.
Fantastic.
So thank you. So thank you very much again for being here with us, Andrea, Dominic, Michael, and also Deborah, Claudia. Thank you very much for helping us to have this meeting today. I also thank Gabriela, who is in the backstage in Lincoln, who is also in the backstage, and everybody for watching us. Thank you very much. And we hope we can come back here and talk, or maybe in presence, we can go to Canada and you guys can come here in the future to talk more closely about all this very nice stuff. Thank you very much again. Have all a nice weekend.
Well, great.
Thank
Thanks so much for having us.
Great.
Thank you.
Thank you. Thank
Have
you.
a nice weekend.
Bye-bye.
Nice meeting
Thank you.
you.
Bye.
Bye-bye.
Missão cumprida.